Good morning, everybody. Uh, Hope you are safe and sound at home. Welcome into AP Bio here at Virtual Learning for the first time this year. Um, Today, we're going to be going over notes from 6.5 and 6.6. I'll make sure that they're put into OneNote here in just a minute because it's going to be hard to fill in the notes if you don't have them with you. But yeah, so we're going to take an opportunity right now to just try to stay caught up with where we're at. 6.5 and 6.6 have some pretty important information that's present. Um, and I have a good feeling that we're going to want to make sure that we know what's going on within this unit or within these two tabs of a unit. And then um, making sure that we're applying it because we're definitely going to need it for when we do our lab. That being said, it's been a weird morning. um, that being said, we just want to make sure that we're following along with what's going on with the notes. Um, I'm going to try to keep this a little more brief. So if you feel like you need to pause the video at any point, feel free. Might make it easier for you when I do I believe there's challenge questions, blue screen questions, like there always is. I'm just going to go through those and then basically I'm going to ask the question and then I'm going to go for the answer maybe like 2 seconds after. And so if you want to wait until the answer, just pause. Or if you need time to think, just pause and then start the video up afterwards. Outside of that, I don't think there's too much else to that we need to know. And so we'll just get right down to it here with our notes for today. The last little bit. Oh, I guess I do have well, my cat teaching assistant was just on my lap, but now it's snack time apparently. Or it's time to go bother my wife. Either way, good job, kitty. So let's get looking at this at our slides here. Okay, so today we're talking regulation of gene expression and cell specialization. Um, when we talk about genes being expressed, that just means that they're being used, they're being turned on or turned off. And so when we express a gene, um, that means that whatever protein is supposed to be being made from that gene is effectively being made, right? And we just went over that process of DNA into RNA, mRNA into the, spliced strand where we're keeping all of the exons together that leaves nucleus. And then we can see that once it gets into the cytoplasm, it's connecting to ribosomes, going through translation in order to make that protein. So when we talk gene expression, we're just saying that a protein is being produced from a specific segment of DNA, right? And so gene expression is going to look a little something like this, right? Prokaryotes and eukaryotes, they have to be able to regulate which genes they express at a given time, right? Your body does the same thing when you are expressing stomach acid genes. It's probably because your body is about to go through digestion. If you are expressing certain hormones, I mean, as growing teenagers, you guys have had this happen for years on end now, where your body is regulating the expression of um, certain hormones throughout your bodies, right? Um, and knowing this expression that this happens, genes can either be turned on in order to express and make a certain protein, or they can be turned off in order to stop making a certain protein. Um, and that can all be based on environmental cues, things that are happening around you, or internal cues where things are happening inside your body. And so your body is then using um, what it knows in order to be able to make the protein that it needs to complete a specific function, right? And on or off, like I had said previously, it refers to whether or not transcription will take place. On means it will, off means that it won't. And the cool thing is that based on genes being expressed, whether they're on or off, allows for cells to be able to perform certain tasks. Because remember, every cell in your body contains your entire genome, right? So it contains all of the DNA in your entire genomic structure, right? Every cell has all of your chromosomes. And so because of that, they're only ever expressing certain genes at a time, right? You don't want skin cells expressing genes for stomach acid as it starts to melt your skin, right? You don't want liver cells that are supposed to be expressing to make proteins to break down toxins in your body to be, um, you know, expressing traits that would be part of the central nervous system, right? The liver doesn't need to transmit signals. It needs to help detoxify the body. And so those cells can specialize based on what genes get turned on and off, right? Now, for bacteria, for our prokaryotes, right? When we do our lab, especially, this is going to be important, but this also happens within um, this can also happen within eukaryotes as well. But it's easier to visualize with bacteria. Um, so there's these things called operons, and they're a group of genes, right? So remember, a gene is just a segment of DNA. And so we have a group of genes that can be turned on and off, and they all happen to be really close together to each other, right? An operon is going to have three main parts. The first part is going to be what's called the promoter, which we had already seen when we talked about RNA, or excuse me, DNA replication, right? There was that promoter region, right? We called it the Tata box, right? Because that's when the DNA is going to start doing its thing. Um, and then directly after the promoter is another segment of DNA called the operator. An operator is more or less an on or off switch. If something is attached to the operator, it's going to be switched off. And if something detaches from the operator, things are going to switch on and proteins can be made. And then the last piece is the gene itself, right? What is the actual protein that needs to be made um, during this process of gene expression, right? So you start with the promoter, you then have the operator, and then you have the genes that's actually being coded for, right? And operons, these whole entire groups, they can be categorized in one of two ways. They can either be what's called repressible or they can be inducible. And we're going to talk about both of those meanings here in just a second. Repressible means that the gene or that operon is in a natural state of being on, right? The expressing those proteins naturally happens. And so you can repress that, right? You can hide it in, you can tuck it away, you can stop it with a certain process or with a certain enzyme or a certain molecule will latch on to the operator and it will turn off that gene pathway, right? So you can have operons that are repressible, where their natural state is to be on, but they can be turned off, and an opposite for inducible, where you have operons that are normally off, but they can be induced in order to start making the protein or the enzyme or whatever molecule is supposed to be made from the gene, right? So repressible, it starts as on, its natural state is to be on and it can be turned off. Inducible, it starts off and it can be turned on, right? And so all of these tend to be part of this bigger process where we see what are called regulatory genes. And regulatory genes their job is to produce a repressor protein that blocks RNA polymerase from transcribing the gene, right? So these regulatory genes are basically like stoplights for genetic expression, right? Once the regulatory gene has created its protein, that repressor is going to lock on to the operon, specifically the oh my gosh, lost track of thought. The operator, right? The repressor is going to lock on to the operator. And once it's locked onto the operator, it's going to prevent any transcription from occurring, right? And so there's specific genes to tell other genes whether or not they can be switched on or off. It's crazy. Gene inception here, right? And there's always these regulatory genes. They always exist, but they're always at low levels, right? They're never really that high because the body tends to need a lot of proteins all the time. Um, the good thing is that any repressor binding, anytime a repressor does bind to that operator, it can be reversed. It's not like a super strong bond forms between the repressor and the operator. It's just probably weak covalent bonds that exist, and so that repressor can be taken away in order to allow for transcription of that gene to occur again. Right. But before we look at examples of where we're seeing regulatory genes do their thing, we want to look really quickly at allosteric enzyme binding. I know we had talked about this back in Unit 2 or Unit 1, where we were talking about what goes on with enzymes. And so we want to take a flashback to see what's going on with allosteric enzymes. All right, so our allosteric activator, um, this is you have an enzyme that is in its inactive form, right? It's turned off. And when the allosteric enzyme binds to the allosteric site, it then opens up the active sites, right? It allows things to occur. And then those active sites remain open, right? So when we talk about it being an activator, it's taking something that's switched off and it's turning it on, right? For the allosteric inhibitor, it's the opposite. We have something that's already on, and once that substrate binds to it, it then switches off, right? And now it isn't able to, um, now it's not able to do whatever it was supposed to do. And this kind of interaction is exactly what's happening in our bodies, especially when it comes to these operons and when your body is regulating what genes it's going to produce. Right. And so when we first take a look, we're going to start with repressible operons. So operons that are normally turned on all the time, but can be switched off. Right, so here's our example for repressible operons. We have what's called the TRP, or the tryptophan operon. Right, the tryptophan operon in bacteria helps to control the synthesis of tryptophan. Right, so um, it's pretty aptly named TRP operon. And it's going to synthesize this essential amino acid, right? And since it's repressible, transcription of tryptophan is always occurring, right? The bacteria is always going to be making it because it needs tryptophan, right? And it can be switched off by a TRP repressor, right? So normally it's on, but it can be switched off by a tryptophan repressor. I'm just going to turn off my heat here for a second. I think we're gonna get drowned out. Anyway, sorry about that. And so what happens is allosteric enzymes that attach to the operator are going to attach only when tryptophan binds to it, right? And so if the cell makes a bunch of tryptophan, it's got a higher percentage chance that one of those repressors is going to bind to the operator, and then that operator is going to turn off for the synthesis of tryptophan, right? So when too much tryptophan builds up, it's more likely that those tryptophan molecules, floating through the cytoplasm are going to latch on to one of those repressors. And as soon as it latches on to that repressor, it tells, or it more or less is signaling to the bacteria, hey, there's enough tryptophan here. You don't need to make any more. Right. And so it stopped making tryptophan. There's enough floating around. And so tryptophan is going to more likely, right? The likelihood of it binding to um, the likelihood of it binding to a repressor is increased the more molecules there are, right? And that's going to shut off the pathway. And so this looks something like this, right? Tryptophan is present. The TRP repressor is always binding to the operator, right? But in the absence of tryptophan, so now we see that tryptophan pentagon um, is not attached to the repressor. As soon as it's not attached to the repressor anymore, um, we can see that it's taken itself off of the operator, and now you can see that sort of peach blob, which is supposed to represent the RNA polymerase, is now able to transcribe itself across all of those different tryptophan genes. Right, so that is repressible. Inducible is going to be the opposite, right? And a perfect example of this is the lac operon for lactic acid, or not lactic acid, excuse me, lactose or lactase, excuse me. The lac operon, it controls how much lactase or when lactase is made, right? And that we know, hopefully, we remember that lactase, the enzyme that digests sugars that are in milk, hence people being lactose intolerant, can't digest those sugars that are in milk, right? And normally this operon is inducible, right? So it starts in a state of being turned off, right? It's being blocked from doing its task to start. And so what will happen, as opposed to the repressor is automatically binding to the operator, right? It's allosterically active, right? That repressor is binding to the activator, or sorry, not the activator, the operator. And there is an inducer for the lac repressor, right? The repressor is that protein that's binding on. And that inducer is called allolactase, right? Allosteric lactase, allolactase, right? And so once it comes into contact, once it binds to the lac repressor, it's going to deactivate it, right? The opposite was happening with repressible, right? We saw when it binds, it's turning off that pathway, or the same thing was happening with repressible. When it binds, it turns it off. But in this instance, when the lac repressor binds, it's going to turn it off. So it's the opposite action, right? Repressible, when it binds, the pathway gets turned off. Here, when allolactose binds, it allows the pathway to turn on, right? And now genes can be transcribed. Sorry if that was a poor description earlier. Get my stuff together. Right, so here up at the top, we can see RNA polymerase and is number one. Number three is our promoter region, and number two, it tells us is our lac repressor. It's active and it's binded to the operator, right? So expression cannot take place. But here in diagram two, down at the bottom, we can see that one of those pluses, the allolactase, has bound to the lac repressor and it has removed it from the operator, right? Number four is our operator. And as soon as it removes it, now RNA polymerase can go through and it can start creating lactase in order to start breaking down some of these allolactose molecules. All right, quick check. So, like I said, I'm going to put the question up and then wait like 2 seconds and then answer it. If you need more time in order to check through your answers, feel free to just pause the video. All right, so here's our quick check. Question one, compare and contrast the bacterial lac operon and tryptophan operon. So both are going to regulate the transcription of genes. Tryptophan is repressible and lac operon is inducible. We got it right. Question two, what are the three parts of an operon? Answer, the promoter, right? That's going to tell RNA polymerase where to attach. The operator, which is going to be your on-off switch, and then the gene, which is going to be what is the RNA polymerase going to make, right? What's the protein? What's the end goal of all of this? Oh, we got that one right. Right. And now we're gonna switch gears just a little bit um to go more into how genes can be expressed. Um some of this stuff looks a little disjointed, and it is kind of because it's sort of just like specific examples of gene expression. And so some of this might look a little weird, but bear with me. The stuff that's in here is good to know, right? So we know, hopefully, you remember from Unit 4 when we were, or Unit 5 when we were doing stuff with phenotypes and genotypes, right? The phenotype, the physical trait of a cell or organism is determined by a combination of a number of things, right? The genes that are expressed, the level at which they are expressed, right? So if you have a lot of melanin genes being expressed over and over and over, you're going to achieve something that has more melanin, darker skin tones, right? But there's also differences between cell types, and that is what we end up calling differential gene expression, right? And so here is what this looks like. It's eukaryotic gene expression, right? Before we were talking prokaryotic, now we're looking eukaryotic. So eukaryotic gene expression, it's regulated at different stages. So when we had seen the bacteria, we saw the repressor binding to the operon or to the operator, excuse me. And that was either telling the sequence that it could be coded for or that it wouldn't, right? Whether it was inducible or repressible, right? But for eukaryotes, genes can be expressed at different stages of this process of DNA unraveling, transcription occurring, RNA processing, and then even all the way down to where we get into protein, the protein being transcribed, or excuse me, translated at the ribosome, right? And so one thing that can change how easily a gene is expressed, how tightly is the chromatin wound, right? We've got DNA, it winds up around those bundles of histones, right? And if you have a harder time unwinding that DNA, um, you make it harder for transcription to occur, right? So if DNA is tightly wound, it's less accessible for transcription, right? You make it even more challenging for topoisomerase to be able to gradually unwind the DNA and allow it to be either replicated or um, I guess topoisomerase isn't used in RNA transcription. Never mind. Don't forget about topoisomerase for transcription. Um, but yeah, so as chromatin continues to wind up, right, it's creating pressure. It really makes it harder for it to be unwound, and if it's harder to unwind, RNA polymerase cannot bind as easily, right? And so how can this be modified, right? How can the chromatin actually be more or less tightly wound? If the histones go through acetylation, that will start to loosen their DNA, right? It will kind of loosen their bonds. But you can also go through DNA methylation, right, where you're adding methyl groups, and that's going to do the opposite thing, right? So acetylation, chromatin will relax. Methylation, the chromatin is going to really condense and bind together even harder. Right, so that's one way for gene expression to be controlled in eukaryotes. Another way is you can mess with how the initiation of transcription works, right? If chromatin's more accessible, specific transcription factors bind to control elements, right? So things that are going to be controlling this process. If gene expression can be increased, or rather, gene expression can be increased or decreased by binding of activators or repressors. So similar to what was happening in bacteria, you can have activators and repressors um, binding to genes and making it easier or more challenging for transcription to happen. Right? You also have the way that RNA is processed, right? We talked about you get that pre-mRNA strand, and then you cut out all of the introns and splice the exons together, right? It's an alternative splicing of pre-mRNA, right? And that in itself is a way for genes to be expressed differently. Um, Translation initiation, right? So now we're messing with once mRNA has already left the cytoplasm or to go into the cytoplasm and it's combined to a ribosome, right? There can be things that mess with the mRNA, feeding through and binding with the ribosome. Right. And so translation can be activated or repressed by initiation factors, things that are going to prevent it or slow down the process of it attaching to ribosomes. One of those things being these microRNAs or these siRNAs, small interfering RNAs, right? And they can bind to the mRNA and degrade it, or they can block translation from occurring if it clamps onto the ribosome and there's these weird RNAs in place, the ribosome is just going to let it go, and then that gene isn't going to be expressed. Right, so there's these little pieces that can attach to that mRNA strand that's going to maybe even mess up the whole process. Right. And because of that, right, because there's these ways that gene expression can get interfered with or that it can be sped up or slowed down, um, it's what allows eukaryotes to develop specialized cells, right? During embryonic development. So, right, when things are embryos, when you just get that sperm and the egg have just fertilized, right, you get that zygote. Um, Differentiation is already occurring in those early stages of development of eukaryotes. And it doesn't matter if it's for a human, for a chicken, for a sheep, right? This differentiation is going to occur across the board, right? And so cells are going to become specialized in both their structure and their function because of gene expression, right? Genes are going to be controlling what these cells are going to do, right? And we call this morphogenesis. It's the cells themselves are physically changing and adapting and changing shape and changing function, and that is going to be what gives an organism its actual final shape, right? Morpho meaning the form of, and genesis, the beginning, right? The beginning of form, right? And so, how do these cells differentiate during early development? How do they know what to do, right? Some of it is based on the substances in the maternal egg, right? Cytoplasmic determinants, they're called. The egg is going to contain more or less instructions on how to build this organism. Pretty cool. Um, Induction, right? Cell to cell signals can change a gene expression, right? As cells bump into each other, they're able to let each other know whether they should be expressing one thing or another. And both cytoplasmic determinants and induction influence influence pattern formation, right? So, um, what's the pattern behind this? What structure are we trying to build? Because most of the time, like our liver is made up of very specific types of cells that aren't really found anywhere else. And so you form a pattern in order to form specific structures, the lungs, the heart, the pericardium, the sac that goes around the heart, right? The brain, the stomach, all are formed from patterns, right? And it's the body plan for an organism is a patterns. It's cells that are developing in particular ways, right? And we call these homeotic genes, and their job is basically to map out what the body is going to look like, right? And so as cells differentiate, one of the other things that plays a critical role is apoptosis, right? That's programmed cell death. And so apoptosis, programmed cell death, is a good thing. Like if we just continue to develop as a sphere, as an embryo, right, our bodies would just be circular, right? We would just be a circular blob um, as we have developed. But apoptosis has to occur in order for certain structures to take form, right? In order to create separations between our bones, in order to create spaces between our organs, and easily and most specifically, our hands and our feet, right? They would be webbed. We would have webbed fingers and toes if apoptosis didn't occur. We needed like this probably was just a big circular structure at one point. As the cells developed, the ones that were in between these structures were the ones that were programmed to self-terminate. And so you have this, wonderful structure of our digits, um, all because part of the plan, part of those homeotic genes is telling cells, all right, it's your time to go because this structure needs to end up looking like this with these movable digits. Right. So apoptosis, when we think of programmed cell death, it's like, oh, that's sad. But in a way, it's allowing organisms to be shaped that the way that they are, which is super important because that's what we're going to be getting into with unit 7 and 8. We're going to be talking about how traits have um, pushed organisms to allow them to live in certain environments, right? When we talk about natural selection. So that's it for today. That was 6.4 or 6.5, 6.6. I recommend starting on the 6.3 and 6.4 quiz that is in OneNote also. I'll put that up in Warrior Web in a little bit. Shouldn't be too much of a hassle um, to get that back to where it was before. Um, Thank you guys for taking the time to make sure that you're following along with today. I know it's, not super easy because we're virtual, and if you have questions from these notes, um, feel free to we can talk about it when we come back on well, I guess I probably won't see you um, Thursday, but Friday, right? Because today's Wednesday, right? It's a day one. So I won't see you guys tomorrow, but we can talk Friday. But it is important that we stay caught up. And so hopefully these notes today help you stay caught up. Um, my little teaching assistant decided that it was nap time at 9:30 or so, and so she's over here sleeping, taking a little mid-morning snooze, but I can see her ears twitching, and so she's doing okay. I'll see if I can rouse her up here, maybe get her into frame. One sec. I'm able to wrangle my teaching assistant here, although she's pretty grumpy. You're okay. Just say hi. Just say hi. Oh, sorry about that. Here she is, though. What you doing? Good job, kitty. You taught that lesson well. All right, everybody. Peace out. Take it easy.